Namaskar. Hamisha Khush Raho Betiya. My name is Dr. Lily Trivedi and I am from Faculty of Agriculture and Veterinary Sciences. Today I am going to take the subject Anatomy, Ecology and Evolution. Now this is for BSc B.Ed 4th SEM ZBC. Now the session is Organization of Shoot Apex and I am from the University of JVWU. Now to start with my session, we have previously discovered what is meristem and what is the theories of meristem. Now in the theories of meristem, here you see a picture. First let's study what is meristem, what is the growth. The meristem is those cells, they are those cells which are continuously dividing. They are complex cells of a common origin. Now they have the ability to divide and further they change into the permanent tissues which lose the ability to divide. Now we have classified the meristems also on different bases and we have studied their different theories also. How they are dividing on the plane of division. What is their plane of division? And what is the plane on based on what is the function? Based on what is the position in the plant body. So we classify these on the basis of the apical growth or their lateral growth. So we have studied in different lectures. Now we come to the organization of shoot apex. We have studied different theories according to the meristem. The first theory we have studied is apical meristem. Then we have studied the apical cell theory, the histogen theory. In the apical cell theory, we have studied that there is a single cell present. It is apical cell and occurs, this type of theory is applicable only for lower like algae and few pteridophytes. Majority of bryophytes, pteridophytes and algae. Now this single apical cell theory is applicable for those. We also have studied the histogen theory in which we have the three layers, that is the dermatogen, the peribulum and the pleurome. Then we have studied the tunica corpus theory in which the cells are in two layers, the tunica and the corpus, where the tunica forms the epidermis and corpus forms the different layers. Now, after this, we have studied the histogenic layer theory. The histogenic layer theory and the cytohistological zones are there present. We have also the region of shoot apex in which there is the different types of regions are present, and here I am showing you some pictures. Now this is the first picture which shows the apical bud or the bud scale, the axillary buds, the leaf scar. Now this is the 1 year growth only, and in the leaf scar there is the bud scar also seen. Now in the 2-year-old growth we see the node and the internodes in the axillary which is formed near the shoot tip. Now the previous 2 years growth also shows leaf scar and the stem is thicker, and before that there is 3 years old, that is there is leaf scars are present. Now here you can see the organization of the shoot apex. The outer is the tunica layer and it consists of more peripheral cells and which divide anticlinally. Now what do you mean by anticlinally? It means perpendicular to the surface of the shoot apex. The corpus which lies below the tunica has initially a single layer and it is dividing anticlinally as well as periclinally, that is parallel to the surface of the shoot apex. Now we see that in the basic radial organization of meristem, there is a central zone. Now that central zone, here you can see the shoot apical meristem. Here we have the central zone. Now it includes both tunica and corpus. Now this tunica and corpus cells, the apical initial cells are self-renewing and undifferentiated. The cells divide in very infrequently. Infrequently means not very frequent, and they contribute to the peripheral zone. Now we also have the peripheral zone. Now this peripheral zone, now this is the central zone, and we have the peripheral zone. Peripheral zone includes both tunica and corpus. Both the tunica and corpus cells are present. The rate of division is higher and it contributes to the organ formation like the leaves, inflorescence, meristem and floral meristem. Then we have the rib meristem that occurs beneath the central zone, and it is the region of mitotic activity. Now it gives rise to the interior cells of the stem. Then we have the layer organization. Now what is layer organization? The shoot apical meristem is usually dome shaped. As we all know, it is dome shaped and have the tunica and the corpus. The tunica are the two layers which contribute, one in monocot and two in dicot, and it occurs primarily in the anticlinal plane. And corpus is the mass of cells in the center, and the division is in all planes. Then we have, as earlier we have studied then we have some other, what is the development of the other parts of the shoot. Now for the other parts of the shoot, we know that the terminal bud is formed by the apical meristem, and in which the dome shape is having all the three layers, the protoderm, the ground tissue and the procambium. Now the primordia are growing at the edges and are growing to form leaves. The meristematic cells at the base of live primordia leaves develop into axillary buds, and they further form branches in the later development. Now the bud formation, now within the bud, the leaf primordia here at the top is crowded close together because the internodes are very short. So here we have the apical bud formation. As for the secondary growth, which is occurring here, it is produced by it occurs mainly in gymnosperm and most dicot and angiosperms and monocot. Now this is due to the secondary meristem, and it occurs on the signal of growth hormones. Now we also have some various types of vegetative shoot apices. Now these shoot apices, as you know, are due to the monoplex, simplex and duplex type. The monoplex is usually very common in the ferns, and there is only one kind of division. That is the anticlinal division. In the simplex type, there is both anticlinal and periclinal type. The monoplex is found in ferns and the simplex is found in gymnosperms. Now we have the duplex type that is found in angiosperms, and it has both anticlinal and periclinal, and there is two layers of anticlinal division. So we have the most common type in angiosperm is the duplex type of vegetative growth of the shoot apex. So I hope you found this lecture informative, and I have taken some of the pictures and references. These are the references from where I have taken my pictures, and so this session is powered by digital version 2.0 of Jyoti Vidyapeeth. And students, I hope you found this lecture interesting. And if you have any queries, you can give in the comment box. Thank you very much. We shall meet again very soon.